Uh, Hi, I'm Struan Grant. I'm a professor in the two departments of genetics and pediatrics at the Perelman School of Medicine at the University of Pennsylvania, and I'm going to talk about epigenetic approaches to variant gene mapping. To give you a very brief overview of what I'll be talking about, is I and my team are involved in harnessing what has been found by GWAS um, and what hasn't been found by GWAS. And in order to find out more about the piece that we don't know from directly at GWAS, we've been using epigenetic approaches. And I'm going to describe really my approach through a number of disease examples, principally obesity, bone, diabetes, sleep, and lupus. This is really how I see the progress in common complex trait genetics. There's been a number of eras that have predated where we are today in terms of trying to uncover our understanding of the genomics of complex traits. And there's really been different key players in each era. So when I was doing my PhD back in the 90s, everybody was interested in candidate genes. So nominate a gene and then study it in depth in the lab. And so that was really the domain of molecular biologists. Then we had opportunities to scan the whole genome in a non-hypothesis approach. And that was principally through family studies or linkage studies at that time. And that was really the domain of statisticians. But then around 2005, 2006, GWAS came along, and also using the same platform, you could look at copy number variation. You needed very large sample sizes to do that. And as you'll have heard in the previous lectures to this, GWAS has been very powerful. It has really been the domain of clinicians because we've been very dependent on our clinical collaborators to collect very large sample sizes. As these collections grew up and we went beyond just arrays, we could do exome and whole genome sequencing and get more and more information. That meant we've got big data. That really became the domain of bioinformaticians. But the whole point of this is to collate in a non-hypothesis way a whole bunch of data to really reveal key insights into biology, much better than the sort of candidate gene era where we were sort of estimating what was going on based on known biology. Here, we're getting new insights into biology that is very robust and consistent. And so that means you can go into the lab with greater confidence to pursue leads coming out of these studies. And so I would argue that the characterization step funneled through all these different approaches in the past, it'll again be up to the wet lab molecular biologist to try and characterize and follow up these very robust leads coming from these large-scale studies. In the genetics piece, I've been involved at the Children's Hospital of Philadelphia with the Center for Applied Genomics, and I came in 2006 with this guy in the picture, Hauken Haukenerson, and our mission at that time was to genotype over 100,000 samples from children and across multiple ethnicities as represented in Philadelphia, and marrying that up with the electronic health record in a de-identified manner, we could get novel genetic insights into pediatric disease. This was all made possible through automation and informatics. And then during that era, we had a lot of success, given that we had the largest pediatric collection at that time, and we led GWASs in that pediatric space. We had GWASs as shown here for these different traits, including neuroblastoma, asthma, and autism. But my area of interest, and really the prism I'm going to talk through what I've been doing in terms of trying to understand function of genetic variants, is in the domain around type 2 diabetes. And my sort of niche area, my unique angle on this is looking at the pediatric route to that disease. And there are a number of ways that can drive risk of type 2 diabetes in the pediatric setting. Principally childhood obesity. If you're obese as a child, you're more likely to be obese as an adult, whereas the converse is not necessarily true. Birth weight, if you've got an abnormal birth weight, either too high or too low, you're more likely to have type 2 diabetes in later life. We're seeing increased risk of type 2 diabetes in kids, partly to do with the obesity epidemic, and that drives into type 2 diabetes risk. And there are other types of diabetes, which I won't touch on, but obviously type 1 diabetes, MODY, and those sort of things. There's some orthogonal traits I'm interested in this context as well. I've got increasingly interested in sleep. If you have bad sleep, you're more increased risk of obesity and diabetes, and vice versa. Diabetes and obesity increase your sleep dysregulation. I'm also interested in the context of bone, and especially bone loading and the endocrine components of bone in relation to these traits as well. So this is really the prism I'm going to look through, that very much that endocrine prism. And increasingly with that Center for Applied Genomics I mentioned, so I was in the tenure track, I had to show independence, so I left the center a few years later and started a variant to function focused in my own lab. But whilst working still with the Center for Applied Genomics, we were lucky enough to lead a couple of international efforts. And just to give you a flavor of some of the GWASs I was involved in leading, this is one birth weight from over to 12 years ago now. But what was interesting about birth weight was that we found a bunch of loci, and we've done subsequent iterations of birth weight, and we found more and more loci. And what's really becoming clear is that many of these are known to us GWAS geeks in the context of adult traits, but it seems like they're involved in pediatric traits. So for instance, these two loci, it does appear that even though they're associated with type 2 diabetes, maybe the principal trait is birth weight. This one's an adult blood pressure locus. These two are adult height loci. So there's that pediatric component, these early life factors. Also been leading for the same consortium, the EGG Consortium for Childhood Obesity, and we found a number of hits that had already been reported in adults. These loci indicated in black, plus we found additional loci in red. What was interesting, though, is that we were able to rediscover some of these loci using a sample size 10 times less than was required in adults, suggesting that there's more of a genetic signature to this trait in kids, probably because there's been less of an exposure to the environment, period. So if they're presenting with obesity, it's more likely to be due to genetic factors. I'm going to come back to that plot in a few minutes, but let me orientate you on what I've been trying to do with respect to understanding the function of these loci. And really the way the field stands at the moment is we're not clear on a number of things with respect to these signals. The problem with GWAS, although it's easy to do in terms of just getting DNA from blood, that's all we have. We have DNA from blood, and we have variants that are overrepresented in patients versus controls. The problem is we don't know what the underlying causal variant is. We don't know what the function of that underlying causal variant is. And it's typically non-coding. So it's in some sort of cis regulatory element upstream or downstream from a gene. And we don't know which gene it's controlling. So what is the regulatory element and what is the effector gene at the end of that connection? We also don't know which cell type these factors are expressing themselves. Maybe the cis regulatory element only works in a particular cell setting, let's say the brain in one context. But once you've established all those, then one could go and do with greater confidence gene function analysis in a particular context. But there are a number of challenges for this follow-up. So coming back to that obesity plot, I just want to bring your attention to the top signal in childhood obesity, and it's also the top signal in adult obesity, and it's the FTO locus. So there was a lot of excitement when FTO was first reported by a British group, and the idea was that if you targeted FTO, perhaps that could be a great way of preventing obesity or treating obesity. So a huge fanfare for FTO. However, around this middle of the last decade, these three papers in the corner here came out, really studying FTO in some detail and really asking the specific question, Is it FTO? So this is where the GWAS signal resides. And as you can see, these little green bars and this UCSC browser plot are all consistent between studies showing that there's something going on in this intronic non-coding region embedded within FTO. And what these papers did in various different ways is asked, what genes in the neighborhood is it regulating? And there was very consistent evidence that if you look in adipose, the gene on the right, IRX3, seems to be the causal gene. So you perturb that green area, you get IRX3. The New England Journal paper listed here from MIT went one step further and said, in addition to IRX3, it looks like it's also IRX5. The final paper listed here actually looked in the brain, and when you look in that context, it suggested the gene on the left. So there seemed to be a cellular context specificity, and it seemed like the effector gene varies. What is exciting about this, from an optimistic point of view, is for the price of one signal, you're getting multiple targets. If you're taking a pessimistic point of view, this is complex, and we have to figure this out. And I think the field is now coming to the point that it is complex and that there are sometimes multiple effector genes at the locus, and it isn't always necessarily the nearest gene or even the gene in which the signal is embedded. Because they're non-coding, all bets are off in terms of what that non-coding region is actually regulating. So I was definitely very inspired by that, and was interested by the physical approaches that they used, which I will talk about in a minute. And to really summarize what I'm saying is, you've got this non-coding variant, which can be sitting between genes or within an intron. And so the question is that non-coding variant that's been found to be associated with disease, in fact, affecting the nearest gene, or is it affecting a gene further away? And so there really are two approaches to this largely. There's the physical epigenetic approach, and of course there's EQTLs, which I won't cover to any degree, but they give us slightly different levels of information, and I'll talk about that a bit later. But what we're focused on is looking at that physical interaction, that genome folding, that looping between the variant and a gene promoter. And what another useful information that comes from epigenetics is the realization in about the last decade that there are TADs, topologically associating domains, where the genome folds on itself really in a series of blocks. This is largely tissue independent. So TAD's boundaries are pretty much the same, irrespective of the cell type you look at. That's a convenient fact in that it can help us set the search space in which we're looking for our causal gene. So in this cartoon example, I've got variant, the variant that I'm interested in, this red star, let's say it's the causal variant, and that the causal gene is actually that gene indicated in red. If we were to take the standard approach, which is to name the locus after the nearest gene, we'd actually be working on the wrong gene. If we were to take an arbitrary window, looking, I don't know, 100 kb either side, we'd still miss the gene. Whereas if we consider the TAD structure, where a lot of looping happens, housed within this TAD, then in this long range example, where we've got the variant on one side of the TAD and the gene on the other side of the TAD, we'd actually have it. It would actually be included in our considerations. And so it would be part of the gene list that we'd be interested in and would indeed be captured. So to give you an example of this is type 2 diabetes, and a locus I've been very interested in for some time is the TCF7L2 locus, which is the main hit in GWASs of type 2 diabetes. And I was fortunate enough to have reported it first in 2006. And so as you can imagine, I'm continuing to be very interested in TCF7L2. And if we look at the TAD harboring TCF7L2, there's a number of genes in there. And the reason I wanted to explore this further is that TCF7L2 is a kind of ubiquitously expressed transcription factor, may not be the most therapeutically attractive, but there are other genes in there that could be more therapeutically tractable and worth pursuing. But of course, we'd have to establish if they are involved. And to put it very briefly, what we did was we perturbed the SNP that is widely recognized as causal in this gene, which is in the fourth intron. And when we perturbed it in a given cellular setting, we found that it did affect the expression of TCF7L2. So we confirmed it is TCF7L2, but we also saw an effect on a gene 3 genes away called ACSL5. That's an enzyme, much more therapeutically attractive, perhaps more therapeutically tractable. And in fact, the knockout mouse already existed at Tufts for other reasons. And indeed, that mouse is diabetic. So we think there is a second gene operating at this locus. This energized us to do this further and do it at larger scale. And so by engaging our own institution, Children's Hospital of Philadelphia, we started an operation around spatial and functional genomics. And the basic concept is as follows. So in these 4 panels, it's really our pipeline. So on the left here is what we do when we first get a GWAS. So the sentinel SNP is that top SNP at the top of the tip of the spire in a Manhattan plot for a GWAS. Of course, it's probably not causal, and we also have to therefore consider all SNPs in linkage disequilibrium with the sentinel SNP, the co-travelers to the generations, if you will. And so by looking at all the proxy variants as being possibly causal, we take that list of SNPs and then impose attack-seq data on it for a given cell type. And so if the SNP is open, it is more likely to be causal because it's in some sort of regulatory element that is open in that particular cellular state. So we shortlist the SNP list to those that are just open, and then we put a second constraint on, which is, are any of those open SNPs being contacted by a gene promoter physically? And we do that through chromatin conformation capture. What that does then is shortlists our set of SNPs to a manageable number at a given locus, and also we have a nominated gene. Then by having those shortlists, it gives us a manageable number of leads to then functionally follow up in whatever cellular setting we've elected to do this in. There are various types of chromatin conformation capture approaches. There's Hi-C, there's Hi-Chip, there's Capture-C. And we first started off with Capture-C because it was highly scalable. You could design where you wanted to design your probes rather than doing every interaction in the genome, which is Hi-C. We wanted to put the focus on promoters. So if we had a SNP of interest, rather than putting probes in the SNP, we put probes in the promoter and say which promoter physically goes to that SNP. And so we had a one-stop shop where we could generate this list of promoter interactions for a given cell type and then see which SNPs are contacted by which promoters. And so Alessandro Casey, who was in my group and is now in faculty at Penn, designed this Capture-C approach where we put a, well, we didn't design Capture-C, it came from the University of Oxford, but we leveraged this technology and made this design where we put in probes for every promoter of the coding genome, but also all non-coding species that had been discovered up to that date. We also designed it around the restriction enzyme DPN2, which is a frequent cutter. What that means is we're dicing up the genome into very small chunks, so we can get very high resolution interactions. A lot of Hi-C at that time was done with a much more infrequent cutter, so you were looking at interactions between large chunks, and so you didn't have the granularity we were looking for. So we had deep sequencing at promoters, the regions we were interested in. We also had a very fine resolution, and then we just did very deep sequencing on the NovaSeq, which then gave us our maps. And just to emphasize this idea of resolution, comparing against the standard HIN3 6 cutter Hi-C at that time compared to promoter Capture-C that we were doing, we found that the DPN2 may not have quite the same range, but it does go to about the distance of a standard TAD, but also it gets us around the myopia of infrequent cutter, in that if you see these loops below, when we've got a fine grain, what's going on near the bait region or near the probe region, we can get very close interactions, whereas we wouldn't see that level of interaction with the high C. The other key thing is, of course, we don't have the same range at the 4 cutter, but we can collapse bioinformatically down our fragments into larger fragments, and so that we can also get that range. So by dialing up and down, we can get close range and further range as well. So this is why we were very interested in doing the 4 cutter. And we were fortunate enough for the Children's Hospital of Philadelphia to embed a NovaSeq in our lab for a few years to be able to generate this in many, many different cell types. So the first disease that we elected to go with was lupus. And the reason for that was my fellow director of the center, Andrew Wells, who's an immunologist, said, let's do it in lupus because kids are frequently getting tonsillectomies at CHOP, so we can get sorted T follicular helper cells from those, and they are the primary candidate cell type for conferring risk of lupus. So it was a good, robust cell model in which to first apply this capture-C approach. So what we did was this is an example of that in TFH cells. I bring your attention to the red loops here. There was a lot of excitement about the LPP locus in lupus. It's a novel gene. It seemed to be new biology behind lupus. However, the proxy SNP that we found indicated here, which is the one that fulfilled our criteria of being open and contacted by a gene promoter, actually pointed to a gene on the other side of the TAD, BCL6. So we think it is not LPP, rather it's BCL6. And the key thing is BCL6 is well known to both lupus biologists and TFH biologists. So in effect, this is known biology. And so there's nothing new here, except that we've found a new regulatory element harboring a SNP embedded within LPP. But our conclusion was the effector gene was BCL6. Andrew found it hilarious that us GWASers, and he's not a GWASer, he thought it was hilarious that we named these loci after the nearest genes. And so he took the nearest gene nomenclature for all these lupus hits and did pathway analysis and found that only 2 of the top 10 pathways are relevant to TFH biology. However, when you take our gene lists, which are led to us through this chromatin interaction analysis, 10 for 10 pathways, top 10 pathways, are relevant to TFH biology. Now, of course, we did it in TFH cells, so there is that, but we did this with just DNA-based hits derived from blood in GWAS. So there was no cellular context for the original hits. What it does tell us is that there's a lot of known biology here. And so what it allows us is to triage away the known biology and just work on the relative minority of genes that are not known and are novel in the field, and where we can put some emphasis. And in fact, there's a couple of genes that came out of this that never been seen in the context of lupus before, had existing compounds, and now we're working on trying to reposition and repurpose those compounds in the context of lupus. Another example I'd like to give is sleep. And we again did the same thing. We took an insomnia GWAS, which I was not involved with at all, but we just took the published hits and we wanted to find the causal genes. And the way we did that was our usual looping approach. But this time we would take our candidate genes and knock them out in flies, and anything that came out positive in flies, we could then check in fish. And the reason we could do this in model organisms is sleep is highly conserved. Everything basically needs to sleep. Sleep is non-negotiable. We all need to sleep. Despite sleeping in the cave in the old days and avoiding and not wanting to be eaten by some wild animal, you still had to sleep. So you were still vulnerable, but evolution did not get rid of sleep. So for those reasons, we felt this was a relevant model for something as conserved as sleep. And in this case, we saw when we took the orthologs that were available in flies and knocked them down, we found that there were genes that did show extremes in activity change. Of course, this is very cross-species analysis between human and fly, so we don't expect every human gene to do something in fly, but we did see these genes do something. And so the red genes on the left of the plot are those where there was reduced sleep time. So these are sort of hyper-caffeinated flies, if you will. And then on the right, we have long sleepers. One of the first genes that came out of the screen was PIGQ, as indicated here. And that was the gene that we first really functionalized and had a head start on. And so, and I didn't include a slide, but we also knocked out PIGQ in fish. And we also got sleep dysregulation in fish. They were also long sleepers in fish. And so that motivated us to go back to the human model. And in this case, we had 3 SNPs in very strong LD with each other in fairly close proximity. We could not ascertain with our approach which of those 3 SNPs are causal. So Shilpa Sonte and my group did luciferase assays for each of those 3 SNPs and asked which ones affect expression differences in a reporter. And we concluded it was this. So by having a pipeline where we were convinced the PIGQ was involved in sleep, it allowed us to go back greater confidence into the lab, work in the relatively more challenging human situation to nail the causal variant. Switching over again to hypothalamus, which I guess is sort of relevant to sleep as well. We were interested in hypothalamus because it's related to many sort of endocrine traits, endocrine-related traits, even though it's in the brain. And in this case, we could do something slightly different. We're trying to take the guesswork out of what trait to study in the hypothalamus. So let me explain. What we did was we take a whole bunch of GWAS summary statistics that were publicly available and integrated them with a hypothalamic model system data set, where we had that at 3 different differential stages. We had at the embryonic stem cell state, the hypothalamic progenitor, and then the hypothalamic neuron, all done in a dish. And where you see an asterisk is where there is a significant enrichment of GWAS hits in our features. So really our conclusion was that age of menarche, bipolar disease, body mass index, height, depression, and sleep traits were enriched in at least one of these cellular states. So as such, we were then motivated to say, okay, given we see that overall global enrichment of genetics for these traits in the cellular setting, we can then go and do variant to gene mapping with our data sets for these traits and do functional follow-up. And this is what we did in the context of childhood obesity. And we had this recent paper from Sheridan Littleton, who's just graduated with her PhD and is now at the University of Cambridge. And she decided to put her energy into one lead that we had at the FAME2 lockets. And the reason we were interested in FAME2 was that if you look at childhood obesity GWAS and compare it with adult GWAS, as I said earlier, they're quite similar. These purple genes have received a lot of focus from the adult studies because they're big hits. Some monogenic genes come up where there's this other flavor of variant, the common variant as well, but they're well-established genes. But in amongst that mix was FAME2, and the interesting thing about FAME2 was that it's lower down the ranks in adults than it is in kids. And in fact, we have observed that the FAME2 signal is stronger in kids than it is in adults. So it seems to be a key early determinator of obesity in children and almost a discriminator between adult obesity and childhood obesity. The key thing is, because it hadn't come up so robustly in adults, it had not received the same level of attention. And so we decided to focus in on that. And it was very fortuitous that when one does the transancestral fine mapping, which is what we reported about 5 years ago, there was really a very strong posterior probability that RS7132908 was the causal variant. And by doing Bayesian fine mapping, the 95% credible set, again, the same SNP was very highly likely to be causal. Imposing a tax-seq from data sets that we have now in over 50 cell types, we saw very consistent evidence of open chromatin across the same SNP. So this cumulative evidence of this being causal is there. And so Sheridan committed to trying to understand this SNP further. The key thing is it sits in the 3' untranslated region of FAME2. Question was, does it actually control FAME2? And in fact, our looping data really did suggest that this 3' UTR region was interacting with the promoter of FAME2. There's also evidence that the brain is really crucial to the onset of obesity. Looking at the genes around GWAS loci for adult obesity, it's been previously reported that there really is an enrichment in the CNS, as opposed to if you look at waist-hip ratio, which is another way of looking at adiposity, that points to the adipocyte. But we were interested in BMI and the CNS. And so given we saw this with FAME2, we pursued the hypothesis that FAME2 was conferring its effect through the hypothalamus. We also saw the looping in astrocytes and a primary astrocyte line. And given the hypothalamus is not for the faint of heart to study because it's highly challenging tissue to get hold of and also to develop in an IPS or ESC setting. So the astrocytes proved an easier setting in which to sort of do the initial studies in. So to convince herself, Sheridan created the luciferase construct, a bit like the PIGQ I mentioned earlier, bringing in the promoter and the risk or protective variant, and saw that, in fact, when you introduced the promoter, there was an increase in, when you brought in the region containing the non-risk variant, there was a large fold increase in the expression of the luciferase reporter, whereas the risk variant led to a reduction. So that really was compelling through multiple replicates that there was a physical effect of having this region containing the variant. She also worked with our IPS core to have on the same genetic background, the only thing that we changed was that one SNP. So having the risk and non-risk allele in the same ESC line, and then differentiating it to hypothalamic neurons. And what was found very interesting is when you do that differentiation, you actually get a mixed population of cells. They're not all pure hypothalamic neurons. There are different sub-cell types, subcategories of cell type. And we found that that distribution was significantly different between the risk and non-risk allele. So the overall hypothesis now is, or the overall observations are, that we consistently saw this risk variant in 3' UTR of FAME2 was interacting with the promoter of FAME2. It was interacting with other genes depending on the cellular context, but what was consistent was FAME2. But we can't rule out that perturbing this variant or perturbing this region isn't affecting other genes in the neighborhood in a more cell-specific manner. But the overall observation was that the distribution of cell populations was different between allele. And we would like to explore this further because what she was looking at was a sort of terminal differentiation at 40 days. FAME2 expression is actually much higher at day 14. And so further experiments are needed to look at day 14 at that peak time, because that's probably where it's exerting its biggest effect in terms of the allelic difference. Pivoting to bone mineral density, we were very interested in the top hit for pediatric bone density at the wrist, the main site of pediatric fracture. Kids get a lot of pediatric fracture, so understanding bone density at that site is very, very intriguing. And so there's a strong hit on chromosome 7. This is also an adult bone hit, but this is really the FTO, if you will, of pediatric bone density at the wrist. And so by doing, again, our same approach, taking the sentinel variant, which did not fulfill our criteria of being open or contacting a gene promoter, but an attack-seek peak nearby, harboring 2 SNPs ending in 00 and 06, as you can see here, were so close to each other, they were within the same attack peak, but they fulfilled the criteria of being open and contacting two gene promoters, in this case, CPED1 and ING3, which is interesting because the main hypothesis for this region in the field is it's Wnt16, which is on the right here. And Wnt is considered, Wnt signaling is considered a key a process for bone density determination. CPED1 had been studied to some degree, but nobody had really considered ING3. So we're interested especially in the lead from ING3, and so we worked with our collaborators at the University of Michigan to knock down the expression of these three genes with RNAi in BMP2 stimulated mesenchymal stem cells, which are then differentiated to an osteoblast model. And measuring alkaline phosphatase, which is a marker of early osteoblast differentiation, and then alizarin red, which is a marker of mineralization because osteoblasts lay down bone. And so to put it in summary, this is what we saw. So you see the control here at the top. So when you add BMP2, it turns blue, which is an indication of alkaline phosphatase and early osteoblast activity. Then it turns red with the case of alizarin red, showing there's mineralization. As you can pretty much clearly see that in our hands, ING3 was doing something when we knocked it down. Now, we can't rule out CPED1 or Wnt16 despite not seeing anything in our assay, because a lot of these effects can be temporal. So our approach is more a case of saying, in our hands, in our cellular state, we saw an effect for ING3. So we're ruling in ING3 as being part of the story at this locus. But it is not for us to rule out the other two genes. But certainly we're saying ING3 is involved. We decided to go further on this, and with funding from the NIH, we're wanting to do this at scale. And particularly in the adult domain, there's been over 500 loci now being reported for bone density. And we thought, well, what happens if we went after all of them? Can we get really major novel insights into that? Now, that requires a slightly different approach in that if we've got a GWAS signal and a proxy, and we think it's in some sort of presumed enhancer, there are transcription factors binding there. It affects conformational change, bringing the enhancer into the proximity of the promoter. How can we determine that after we've done our chromatin capture? Can we use something like CRISPR? So we used a CRISPR crab to repress that enhancer, therefore preventing the transcription factor from binding, and then preventing that conformational change. And by doing that at scale, and going after all the leads that we got from our causal variant to putative causal variant to putative effector gene, we had a list, and we perturbed it using single cell RNA-seq. By looking at the transcriptome from each cell in turn, knowing which cell got which guide, we saw the following. We saw an enrichment of genes when we did the targeting. The interesting thing was that half the genes were not reported from our chromatin capture. It was a different gene in the neighborhood. It was clear that we'd hit some sort of functional causal regulatory element, but the cell state may have been slightly different, but we're seeing a different gene being perturbed. We were very excited about that and also slightly concerned because we're seeing sometimes the right gene or the gene that we nominated with the chromatin capture coming up and sometimes not. The key thing is that when we went and did functional follow-up, again with our Michigan collaborators, vast majority of them checked out with respect to alkaline phosphatase and mineralization. But we're going back now and thinking more about that temporal aspect, about depending on the exact cell state, the exact passage number, what genes are actually at play at this neighborhood. So they're clear that there's some temporality happening at these loci. But we're very excited at the fact that we hit quite consistent key regulatory elements harboring causal variants. Coming back to type 2 diabetes, we were lucky to get NIH funding to do variant gene mapping for type 2 diabetes. And there's really two sectors to this funding. There's the computational piece, which was our approach using chromatin capture to nominate variants in genes. And also Casey Brown, who obviously this lecture series is dedicated to, and was in early days before his unfortunate passing, was involved in the EQTL nomination. And so by these two different approaches to nominate causal genes, we could then hand them over to our functional colleagues who are working in liver, islet, adipose, muscle settings to functionalize the leads. And also what's been exciting is there was 2 grants awarded, and our colleagues at Stanford, Michigan, UNC, and the Broad, it's been exciting sort of following up these leads together. And some of the first fruits of this was, again, doing our in this case, doing high resolution Hi-C, given that Hi-C is now becoming more and more high resolution as time goes by, that we're kind of pivoting more to that and deriving our motor leads from that if we do very deep sequencing. And again, using that LD score regression approach, which is sort of saying where is the enrichment before we go and do functional variant and functional gene analyses. In this case, the red represents significant enrichment. And so by taking a series of type 2 diabetes related trait GWASs and integrating it this time with acinar, alpha, and beta cells sorted from the pancreas, we could see where there was enrichment. And what was satisfying was that type 2 diabetes, not surprisingly, was enriched in the beta cell, also in the alpha cell, but not in the acinar cell. Conversely, pancreatic cancer has been largely hypothesized to be driven mostly by the acinar cells, and our data supports that. What was interesting is that sleep, whatever reason, seems to be significant across all 3 cell types. But that allowed us to then go ahead and do more variance gene mapping and characterizing of genes. So this is becoming more and more powerful for us, and as we accrue more and more data sets, where we're nearly at 60 cell types, where we've done a consistent chromatin capture, attack-seq, and RNA-seq analysis, our trifecta, if you will, we can then now take some of the guesswork out and say, if we've got interesting GWAS, where is it enriched before we go and do functional work? Let me give you an example, almost a positive control. In this case, we're looking at SNPs that are open and contacted by a gene, an OCR, open chromatin region, and we looked at across 16 autoimmune trait GWASs. So if I orientate you on the slide, down the left, the first panel is immune cells, then it's metabolic related cell types, then it's neuron, and then it's other, if you will. The colors represent different coverage. Some have got better coverage than others, but they're all sufficient coverage. And if you'll notice that it's the immune panel that shows the greatest enrichment. And again, the asterisk represents significant enrichment with this LD score regression. So it's nice to see that there is significant enrichment in the cell types you'd expect. In some cases, like Graves' disease, which is the 6th disease along, we're also seeing enrichment in other cell types we didn't expect and may be worth pursuing. And so now we can do an LD score regression in any GWAS and look at those leads. And just sort of wrapping up, we've been applying this on a number of fronts. Alzheimer's disease has been one, and Alexander Casey from my group now on faculty has been working with me on this, and we have a grant together. And at that time, Casey Brown also got an NIH award to study Alzheimer's. And so when this graduate student, Beth Burton, came in, she decided to be co-mentored by myself and Casey. And so Beth's been doing great, and hopefully will be defending soon, and got an F31 award to do this variance gene mapping with both approaches in the context of Alzheimer's. So really, there are an arsenal of approaches now, and I hopefully I've convinced you that chromatin capture and luciferase and attack-seq are one powerful way in which to shed light on GWAS loci. And I think we're getting more attention with this approach, given there are some drawbacks to GWAS and EQTLs. And this paper that came out in Nature Genetics from Jonathan Pritchard's group says something pretty key to our understanding of EQTLs. And there's a key line in this abstract that genes near GWAS hits are enriched in key functional annotations, are under strong selective constraint, and have complex regulatory landscapes across different cell types. Whereas genes near EQTLs are depleted of most functional annotations, show relaxed constraint, and have similar regulatory landscapes. What that's saying, based on their data, is that EQTLs and GWAS pick up very different things. Now, if you get a co-localization between an EQTL and a GWAS, that's great, and it gives you a very strong lead in which to persist. But what it does mean is that a lot of the time, co-localization is not going to give you a lead. And I think chromatin capture is a good adjunct to EQTL analyses. And that's where, when Casey and I worked together, and Casey was the first to acknowledge that there was some drawbacks to EQTLs, even though he was a great leader of GTEx, he was very into the idea of combining our data sets, really getting a confluence of evidence from both orthogonal approaches. And again, working with Casey, again, we contributed our respective data sets in the context of a GWAS that was being published for NAFLD, and we saw nominated genes with our variant to gene approaches, and that was incorporated into the initial GWAS. And the last paper that Casey published before he died, I'm sure there's more papers to come, but this is the latest one in PubMed. This was another idea of providing data sets to do variant to gene mapping, in this case for polyunsaturated fatty acids, and being looked at ethnicities which haven't been studied to such a high degree until now. And so we were pleased to be able to put context for those populations. And I'd just like to leave you with one final thought, that this is the latest GWAS of height, and it's really the first GWAS where it's effectively solved with respect to common variation for common differences in height. And the idea being a saturation GWAS, where the conclusion was that if you were to do an even larger GWAS, you wouldn't find anything more, at least in European ancestry. What that says is that there's over 12,000 loci covering about 21% of the human genome. That's a lot of signals. It also means there's a lot of variant gene mapping needing to be done. And I think the EQTL field, largely stimulated by what Casey's seminal work did, and what we're doing with chromatin capture, of which Casey was a huge advocate for, I think we've got our work cut out for us to try and elucidate these many variants coming out in complex traits. And I wouldn't be surprised as we solve other traits with respect to the full genetic characterization of common variation, that we're going to have to really scale these approaches. So with that, I'd like to acknowledge everybody in my group and all my collaborators, in particular Casey, of which this lecture series is named after. And it was an honor to work with him throughout the years, and particularly to co-mentor Beth Burton. And laterally, we were also co-mentoring Max Dudek, who you can see in the second picture, second from the front on the right here. And with that, I will close my talk. Thank you very much.